Hi, welcome back. In this biology lab, we will be investigating osmosis. Now, before we do that, let's talk about diffusion really quick. Diffusion is a natural law of physics, which just tells us that particles go from where they are high in concentration to where they're low in concentration. This requires no energy. The random motions of particles spread themselves out until they reach a state of equilibrium, where instead of being concentrated in one area, they diffuse, they spread out into their environment, maximizing their total entropy. Materials diffuse in and out of our bodies and our cells all the time. Oxygen diffuses into our bloodstream in our lungs because it is low in concentration in the bloodstream, but very high in concentration in the air sacs of the lungs. And opposite of that, carbon dioxide diffuses out of our bloodstream because it is relatively high in concentration, but very low in concentration inside of our lungs. Now, normally when we talk about diffusion, we talk about solutes that are dissolved in water. This does happen in gases as well, but in biological systems, pretty much everything we are dealing with is in solution. Your body is mostly water. The cells that make up your body are mostly water. So all of the particles we study are solutes. They are dissolved in water. Now, sometimes you have two unbalanced concentrations, one on one side, one on the other, but the particles cannot move across. The solutes cannot move across. So how is equilibrium reached? Well, we reach equilibrium through osmosis instead. Water is transferred from one side to the other in an attempt to dilute one of the concentrations so that it matches the other concentration. That is what we are going to try to simulate and investigate today. So in order to study osmosis, you need a semi-permeable membrane which will let water go across, but will not let your solute move across. Or if it does let the solute move across, the solute needs to move much more slowly than the water does. And what we have to do that is chicken eggs. I soaked these for 3 days in vinegar. The acetic acid in the vinegar chemically reacts with the calcium carbonate shell and totally dissolves it. And all we're left with is the skin, the very thin membrane that surrounds the chicken eggs. This is going to serve as our model cell. Well, actually, an egg is a cell. This chicken egg technically is one cell, which makes an ostrich egg technically one cell also. Weird. Now, when talking about concentrations, we have 3 different types of concentrations. We always compare the environment to the cell, or the concentration in the environment to the concentration of the cell. A hypertonic solution is one where the environment contains many more dissolved solutes than the cell itself. This causes water to exit the cell in an attempt to dilute the environment to match the concentration of what we see inside the cell. So, for 5 of these eggs, they are going to be placed in a hypertonic solution over here. And to make a hypertonic solution, we're just simply going to add salt to water. This is going to be very hypertonic. Oh yeah. The opposite of a hypertonic solution is a hypotonic solution. A hypotonic solution is one where the environment is very dilute compared to the cell. So in that case, water rushes into the cell to try to dilute the concentration of the cell to match its environment. So the other 5 eggs are just simply going to be placed in filtered water. Now, we can't watch osmosis happen, but we can measure the effect. So what we will do is we will calculate the mass of the eggs before and after diffusion, which I have to do that right now. It's going to take some time, but through the magic of video editing, it should take no time at all for you. Just sit back and enjoy the music. Okay, so we have all of our eggs weighed. I have their mass right here. So we have our initial mass. We're going to compare that to our final mass at the end. We're gonna let these eggs sit for at least an hour so that they have plenty of time for that water to diffuse across the membrane in one direction or the other. So we'll just wait and see what our results are. Okay, so it's been about 2 hours since I put these eggs into these solutions. So what the prediction for what should happen is these eggs are in a hypertonic solution. So what should happen is water from the egg is jam-packed full of a lot of protein, but there's also a significant amount of water. We should see water exited the eggs and they should lose mass because of that. Whereas these eggs, which are in a hypotonic solution, now there's a key thing that we can see here because of that. So if you look at this video, notice that all of these eggs are sitting at the bottom and all of these eggs are floating on top. The density of this water is greater than the eggs themselves, which are full of materials. So that tells us that if the eggs were not floating, these would not be, this solution would not be hypertonic compared to the egg. Whereas the fact that we know these are sinking tells us that this is definitely hypotonic, because as you add solutes, you increase the density of your liquid. So anyways, we should expect these to gain a significant amount of mass because they should have taken on a lot of water. So I'm gonna weigh these and then we'll take a look at the data and see if that is confirmed. Well, this didn't actually go the way I thought it would go. It turns out, here's the data. It turns out that all of my eggs gained a little bit of mass. Now, I've done this lab before using syrup instead of salt, which is extremely thick and viscous and obviously very hypertonic. But syrup is really messy and pretty expensive, and I just didn't have any this year. I poured so much salt, I made this a saturated solution, which means I couldn't dissolve any more salt. Any extra salt I added remained as salt crystals. So it couldn't be dissolved. So I thought that that would do the trick. And the eggs were floating, which told me it was nice and hypertonic. About an hour into the experiment, I took out one egg and it had lost about a couple tenths of a gram in mass. So I thought, okay, everything's going according to plan. But in the end, every single one of these gained, it's very, very tiny, but they gained a little bit of water. Well, they gained a little bit of mass. I'm not 100% sure why that is. An egg is full of protein, so it is very concentrated on the inside, but I still thought we would lose some mass. Now, there is a proof of concept here. It's not all a loss. And in science, sometimes your predictions are wrong. The hypotonic eggs, the eggs that were in pure water, they gained a really significant amount of mass. It appears that diffusion of water, osmosis, occurred in both experimental setups. However, because this solution was much more, well, this solution had dissolved substances, whereas this one did not, the total diffusion, and we could probably assume that the rate of diffusion as well, was much quicker in the hypotonic solution, the eggs in the pure water. So we've still basically proved our concept. The eggs over here must have been in very close to an isotonic solution. Now that was something I didn't mention, but isotonic means one solution is equal in concentration to the other. So they must have been very close. We only had a tiny bit of water, a tiny bit of mass added to all of our eggs in the very salty water, whereas the other solution was definitely hypotonic. So that is our lab, not the results I was looking for exactly, but I still hope you were able to learn from this. I know I did. I'll do it different next time. Thank you for watching.